Welcome to AP Biology. I'm Margaret Evans from Woodbridge, Virginia, home of the Woodbridge Senior High School Vikings, and I will be your instructor for this lesson on topic 1.1, structure of water and hydrogen bonding. So let's get ready to science. What will we learn? In this video, we will learn about the chemical structure of water, how that structure is resulting in something called polarity. How does water's polarity result in cohesion and adhesion through hydrogen bond interactions? What chemical characteristics of water result in its cohesive and adhesive properties? And how do living systems depend on the chemical properties of water? Let's begin. It's important to understand that the subcomponents of biological molecules determine the properties of that molecule. And as it relates to the components of water, water is composed of two elements, oxygen and hydrogen, as you can see from the diagram that just appeared, in a 1:2 ratio respectively. Covalent bonds are the bond types that are involved in a water molecule. Oxygen is sharing electrons with hydrogen. However, that sharing is not done equally because oxygen is more electronegative compared to hydrogen. And this will result in something called polarity. And so I'm going to add a negative sign near the oxygen portion of the molecule, indicating that the electrons are being pulled closer to the oxygen. And then that leaves the hydrogen area more positive. And once again, this dual property of charges on a molecule is referred to as polarity. Since this is a covalent bond that results in polarity, we can also call it a polar covalent bond. Hydrogen bonds are weak interactions between the negative and positive regions of two molecules. And considering that water is polar, then it has the opportunity to participate in forming hydrogen bonds with other water molecules or with other molecules that have a charge. When you have two of the same molecules forming hydrogen bonds between them, we call this cohesion. And an example here would be a water molecule bonding with another water molecule. And as you can see, I have these molecules positioned so that the negative side of one water molecule is positioned near the positive side of the other, and thus they can form a hydrogen bond in between. When you have two different molecules forming hydrogen bonds with each other, this is called adhesion. And so my example here would be an amino acid and a water molecule. And as you can see, the amino acid also has that dual polarity property. And so the negative side of that amino acid will be positioned near the hydrogen's positive side. And a hydrogen bond would be able to form between the two of them as well. Living systems depend upon these properties of water that derive out of its polarity. And so hydrogen bonds between water molecules can result in something called surface tension, which we'll talk about a little bit more in the next slide. And so cohesion, adhesion, and surface tension allow for water to demonstrate additional chemical behaviors or properties known as the emergent properties. And we'll get into looking at several examples of those. Life depends upon these properties of water. And let's take a look at the first one, which would be surface tension. Surface tension is a result of increased hydrogen bond interactions between water molecules only at the surface. And so the classic example would be a water droplet on a penny. And I have a video here that'll show you a leaf floating on the surface tension of water. And how could this be important to living things? Well, leaves of aquatic plants oftentimes rest on the surface of water, allowing them to have more access to sunlight for processes like photosynthesis. The next example we're going to look at is water solvency abilities. Now, I know we're tempted to say that water is a universal solvent, and that is inaccurate. However, water does have very high solvency abilities in its liquid state. So take a look here at this video that shows salt being dissolved in water. Now, why is this an important property that life depends upon? Because organisms must obtain key nutrients from their environment. Considering that living things are made up of 70% water, dissolved materials in the water allow for easy access by cells. Our next property we're going to look at is the ability of ice to float. Now, water's cohesive properties allow for unique bonding interactions in water's solid state, and this makes water as a solid less dense than water as a liquid. And here we can see in the video with the ice being added, we notice that it floats, leaving liquid water underneath. Now, how is this an important property for living things? Well, aquatic organisms are still able to live in water in freezing climates because the water will freeze on the surface, leaving liquid water underneath for those organisms to still thrive. Next, we're going to take a look at water's high heat capacity. Water has the ability to absorb a lot of thermal energy before it changes chemical states. It resists sudden changes in temperature. And so organisms like my friend here, my little fish friend here, lives in a water environment. And so many aquatic organisms like the fish will depend upon this property of water so that they can maintain appropriate temperature regulations of their bodies. Lastly, we're going to take a look at capillary action. Capillary action is a result of the adhesive and cohesive properties of water. And in this video here, you'll see that as I add a thin tube into the liquid automatically goes up into the tube. Now, how is this property important for living things? Well, plants can access water from the soil through this capillary action ability into their roots. So let's practice our skill for this video, which is a describe, in which we're going to use a visual representation to describe characteristics of biological concepts, processes, or models represented visually. So here is our question that I would like for you to consider. So pause your video at this moment, read through that question, and we'll come back in a second to see what your answer is. All right. So we're looking at describing the property of water illustrated by this graph. And so the answer that I have is that the graph shows the water temperature remaining relatively the same as the air temperature increases over the 6-day period. And this is demonstrating water's high heat capacity. Now, if you notice, it is a describe, so you must describe how this visual represents that, opposed to just saying it's representing high heat capacity. And that's the difference between an identify and a describe. And we're also asked to describe how life, such as Micropus salmoides, which is a fish species, would depend upon it. And so once again, the fish's ability to maintain optimum body temperature is dependent upon water's ability to absorb heat and resist those temperature changes. So what should we take away from this video? Once again, we have to understand that the chemical components of water do influence how water interacts, and that the oxygen and the hydrogens within the molecule do result in polarity due to the unequal sharing of electrons, and this results in water's ability to form hydrogen bonds and exhibit some abilities like cohesion and adhesion and surface tension. Always remember, science is not a class, it's a mindset. So set your mind on science and make sure that you continue to take a look at the videos that we offer. Biology out. Glad you've joined me on this virtual journey into topic 1.2, Elements of Life. I'm Margaret Evans, and I will be your instructor for this lesson as we take a look at the chemistry of life. So let's get ready to science. What will we learn? In this video, we will learn about the living systems and how they require a constant input of energy, why living systems must exchange matter with their environment, and what role does carbon, nitrogen, and phosphorus play in the building of biological macromolecules. Living systems do require a constant input of energy to accomplish all of the processes that are necessary to sustain life. And the law of conservation of energy states that energy cannot be created or destroyed, only transformed. Living systems must follow the laws of energy. And we need that energy in order to grow, reproduce, and maintain organization. Oftentimes, the energy that is accessed by living systems is stored in chemical bonds. In this video, we see how this athlete is utilizing the stored energy in the chemical bonds of the food that we eat in order to have his muscles work, play, and do the necessary activities to sustain living cells. Living systems must require an exchange of matter with their environment. The only way that we can get certain atoms and molecules necessary for life is through accessing them in the environment. And so atoms and molecules from the environment are necessary to build important molecules like carbohydrates, proteins, nucleic acids, and lipids. Carbon is used to build all of them. And so in our data table here, we're going to go ahead and check off each of them, as carbon is necessary for the building of all four. Nitrogen is used to build proteins and nucleic acids. And lastly, we have phosphorus, which is used to build nucleic acids and certain lipids, which we'll talk about a little bit further. Looking at an example of how carbon is used to build a macromolecule, carbon has a unique ability to bond with other carbon atoms, creating what's called carbon skeletons. And here are some examples of carbon skeletons. We have carbons connected to each other to form chains, rings, and branches, and other atoms can attach to those carbon skeletons. And this allows for these carbon skeletons to give cells opportunities to build very large and complex molecules. Carbon-containing molecules can be used, as we mentioned before, to store energy for later use. And the carbon-containing molecules can also be used to form basic cell structures like cell membranes. So what should we take away from this lesson? Well, living systems, once again, need a constant input of energy because they have to abide by the laws of the conservation of energy. Atoms and molecules from the environment are necessary to build the important molecules of life. Carbon is used to build all of the macromolecules, and it also assists in storing chemical energy and forming basic cell structures. Nitrogen is used to build proteins and nucleic acids, and phosphorus is used to build nucleic acids and certain lipids. So, life is chemical. Biology is everywhere. You can't escape it, so you might as well enjoy it. Hope you enjoyed my lesson. Biology out. Margaret Evans here to give you a lesson on topic 1.3, Introduction to Macromolecules. Are you with me? Okay, well, let's get ready to science. What will we learn in this video? Well, we will be taking a look at some of the key properties of monomers, what type of bond connects monomers in biological macromolecules, and what are dehydration, synthesis, and hydrolysis reactions. Monomers have very important chemical properties. And what are monomers? Well, monomers are chemical subunits that are used to create polymers. Now, polymer is a term that we use to refer to a macromolecule, a very large molecule that's made up of monomers. And so here in this diagram, we see how the one shape by itself represents that monomer unit, and it's important for us to remember that we're not talking about one atom, but we're talking about a subunit that collectively is referred to as a monomer. And when those monomers are connected, then they can build larger molecules that we call polymers. A covalent bond is formed between two interacting monomers, and so that's how we get those two monomers to connect. Monomers have very specific chemical properties that allow them to interact with one another, and we'll be taking a look at some of the key monomers that make up macromolecules in some of the later slides. It's important to also remember that the polymers are specific to the monomers that they consist of. And so as we take a look at this chart, we see the polymer and we see the monomer. So you can't just take any monomer you want and create any polymer. Specific monomers are used to build specific polymers. One of the key reactions that living cells use to create these macromolecules is something called a dehydration synthesis reaction. And the subcomponents of a water molecule are actually removed from the interacting monomers, which allows for covalent bonds to form between those monomers. The hydrogen and the OH, which are those subcomponents from water, will eventually join together, resulting in water as being one of the byproducts or final products of the reaction, in addition to the polymer. And so here's a video that I'll walk you through that shows you the idea of a dehydration synthesis. So we have our two monomers, and so the water subcomponents are removed, allowing those monomers to connect with one another via a covalent bond. And then the two subcomponents of water join, resulting in a water molecule. So at the end of this, we end up with a water molecule, and we end up with our polymer. Hydrolysis reactions actually are sort of the opposite of a dehydration. This is where we have a polymer and the polymer is being digested, or the bonds between the monomers within those polymers are hydrolyzed, and the covalent bonds are broken during this reaction. In addition to the bonds between the monomers being broken, a water molecule also has to be hydrolyzed back into subcomponents, and those subcomponents will be added at the end of the process to the monomers. So here we have a video showing a hydrolysis reaction. We have our water that's going to be hydrolyzed into two subcomponents, and we have the actual polymer that's hydrolyzed as well, and the subcomponents of water will be added back onto the monomers. And the result would be our two separate monomers. So let's look at an example of a dehydration synthesis, and this one is going to involve creating a carbohydrate. And so carbohydrate monomers have hydroxide and hydrogen atoms attached to them, and when two monomers of a carbohydrate join together, one of them will lose a hydroxide and the other one will lose just a hydrogen on one of those hydroxides. Covalent bond will then be formed between the two monomers in the location where the hydroxide and the hydrogen are lost. And so the diagram shows you the new covalent bond that will be formed. And once again, the hydrogen and the hydroxide will join to form a molecule of water. Another example of dehydration this time will be one that involves the creation of a protein. And so protein monomers are called amino acids, and they have an amino group that has some hydrogens attached to it on one end, what we call the terminus of the molecule, and we have a carboxyl group on the other terminus. And those groups are going to be involved in the dehydration synthesis. And a hydroxide will be lost from a carboxyl on one side of one amino acid, and a hydrogen will be lost from the other. And so here we have the highlighted hydroxide that's going to be lost off of that amino acid, and the hydrogen that would be lost off of the other. A covalent bond will form between where these two have lost their components. And we see the new covalent bond there. And once again, the hydroxide and hydrogen will join together to form a molecule of water. And lastly, for an example, we're going to show a hydrolysis, which is the opposite reaction. And so remember, for hydrolysis, the covalent bonds are broken. And so here we have a protein polymer, and this is the bond that's going to be broken. In addition to breaking that bond, a water molecule will have to be hydrolyzed back into the OH and H, so that once those bonds between the monomers have been broken, the water components can be added back onto them. As we see there, the OH is added there, and then we have the hydrogen added back there, and we end up with separate amino acid monomers. So what should we take away? All monomers contain carbon and are used to build biological macromolecules, and we use covalent bonds to join those monomers together. A dehydration reaction is one that takes monomers and joins them together to create these macromolecules, and a hydrolysis is a reaction that will break apart the macromolecules back into their monomer components while also hydrolyzing water. Biology, biología, sing how, no matter how you say it, it's awesome. Biology out.
Welcome to AP Biology. My name is Paulette Unger. In this lesson, I will discuss with you the directionality of the subunits of carbohydrates and how a change in the subunits of carbohydrates may lead to changes in its structure or function. Carbohydrates comprise linear chains of sugar monomers connected by covalent bonds. Here are two different chemical configurations of a common carbohydrate monomer glucose, in which the only difference is in the direction of the OH group, shown here. So in the top form of glucose, the OH group is pointing down off the carbon, and in the bottom form of glucose, the OH group is angled up. Even small directional changes like this in the components of a molecule can result in functional differences. So for example, when several of the form of glucose shown at the top are joined together to make a carbohydrate, you get starch, which is used for energy storage in plants. However, when the glucose monomers on the bottom are joined together to make a carbohydrate, you get cellulose, which is used for structural support in plants. And here I've highlighted for you the covalent bond, and notice even the difference in the angle orientation of these bonds because both of these polymers are assembled from different monomers. So even the orientation of the covalent bond is different since they're assembled from the different glucose subunits. So although both starch and cellulose are found in plants, each has a different structure due to their assembly from different sugar monomers, and therefore each of these polysaccharides performs a different function for the plant. In terms of the overall shape, carbohydrates can be linear or branched. Here we have three visual representations of polysaccharides. Starch, which is shown in its branch form in this visual, although I do want you to keep in mind that some forms of starch can be linear. We have glycogen, which is branched, and cellulose, which is linear. So let's just talk about starch and glycogen for a minute. Both are assembled from similar forms of glucose, and these glucose monomers are connected by covalent bonds in similar locations and orientations that lead to this branching structure that we see here. Since these two polymers have similar structure, and structure may lead to function, then it might not be too surprising that both starch and glycogen have similar functions in terms of their role in energy storage. Starch is a stored form of sugars and energy in plants, whereas glycogen is a stored form of sugars and energy in animals, such as humans and other vertebrates. Now, in the case of cellulose, it's assembled from a different form of glucose monomer, which was shown in the previous slide, and therefore, when those particular subunits are connected, they form long, linear, unbranched chains. And when these linear strands are arranged side by side, like you see here in this visual, hydrogen bonding can happen between these strands, and these interactions between the cellulose strands will cause the cellulose molecules to cluster together, and therefore they can take on a more structural role. For instance, cellulose is commonly providing strength in plant cell walls. So let's practice the skill of argumentation and making predictions. Recall from the previous lessons that predictions are expected outcomes when certain conditions are present or changed. So let's look at a question having to do with carbohydrates. Here we have figure 1. It shows the test results after an indicator was added to a solution containing a linear carbohydrate A and chemical X. And we also have figure 2, which shows the structure of a carbohydrate B. If we go down to the question and read the stimulus here, a student studying the effects of chemical X on the hydrolysis of carbohydrates sets up an experiment to test the ability of chemical X to hydrolyze carbohydrates found in plants. Chemical X is known to only digest linear carbohydrate polymers. The hydrolysis of a carbohydrate can be observed by using an indicator that is blue and changes orange when the reaction occurs. The student runs a test in which the indicator is added to a colorless solution containing carbohydrate A and chemical X, shown in figure 1. Now, the student plans to test the effect of chemical X on carbohydrate B in figure 2, predict the resulting indicator color, and predict the relative amount of monomers found in the carbohydrate B solution after the indicator was added compared to the amount in the carbohydrate A solution after the indicator was added. So we have two items to make a prediction about in this question. So I'm going to ask you to press pause, study your figure, read the question. You know, when you have a lengthy question like this, it might be good if you were doing this on paper or on your computer, if you wanted to like highlight certain key terms, you know, or put a box around certain content items to help you leading up to your answer. So press pause, and when you have a prediction, hit play and we'll discuss it together. Okay, hopefully you have your prediction handy. So let's see what a good prediction would be for this case. The indicator would be blue, and there will be less monomers found in the carbohydrate B solution compared to the amount in the carbohydrate A solution. All right, so let's take the first part here. Let's go back to our question. Here are some key terms and content items that jump out. We're talking about hydrolysis, which hydrolysis is the breakdown of polymers chemically into smaller subunits, their monomers. Chemical X is known to only digest linear carbohydrates, and the indicator is blue and changes orange when the reaction occurs, when that hydrolysis occurs. So here we have, if we go to figure 1, carbohydrate A was linear, we added the blue indicator, and it changed to orange, which means that chemical X was able to hydrolyze or break down carbohydrate A into monomers. That's why we see that orange color change. If now the student uses carbohydrate B, notice the structure of carbohydrate B is different. It is a branched carbohydrate. And chemical X is only known to digest linear carbohydrates. So one would think that chemical X would not be able to digest the branched carbohydrate B. And so the indicator, when added to that solution, would stay blue because it would only turn orange if that digestion occurred. So that's answers part 1 of our prediction. The other aspect of the prediction is the amount of monomers. Well, if chemical X is not able to digest branched carbohydrates like carbohydrate B, then we would assume and predict that there would be less monomers found in the carbohydrate B solution, since hydrolysis would not have occurred as successfully as it did when it was digesting carbohydrate A, which was linear. So indicator would be blue, and less monomers because the chemical X would not successfully break down the branched carbohydrate B. So that's how you see that structure and the difference in structure come into play, with how these carbohydrates may also function and respond to changes. So here are some key takeaways from this lesson. Carbohydrates comprise linear chains of sugar monomers connected by covalent bonds. Sugar monomers may vary in the direction of some of their components, like I showed you with those two different forms of glucose and those OH groups coming off in different orientations. Depending on the type of sugar monomer used in its formation, a carbohydrate polymer may have a linear or branch structure and can differ in function. Thank you for watching, and I hope this video was helpful.
Welcome to AP Biology, topic 1.4, carbohydrates. I am Linda Loju, coming from you from Plano, Texas. In this daily video number 2, we will discuss the directionality of the subunits of carbohydrates and how a change in the subunit of carbohydrates may lead to changes in structure or function. So what will we learn in this video? We're going to learn 2 things. So number 1, what is the basic subunit of a carbohydrates? And then number 2, how can a change in the subunits of complex carbohydrates, or what we call polysaccharides, lead to changes in structure or function? So in order to talk about carbohydrates, we have to look at their monosaccharides, so the monomer, right? Remember, the monomer is the building block, the basic subunit, the smallest subunit that can be there in order to be put together to build larger things. So for a carbohydrate, we have, again, our monosaccharides, our one simple sugar subunit that when put together, 2 or more sugar subunits, as you see in the picture, we can call that a polysaccharide or a complex carbohydrate. Looking at now how monosaccharides can form those polysaccharides, those complex carbohydrates, these polymers, we can look at each subunit. So if we have a subunit of glucose, as you can see in the image, when we want to connect the 2 glucose subunits together, we are going to lose a water. That is that dehydration synthesis. So making that covalent bond between the 2 subunits is a dehydration synthesis reaction where water is removed in order to connect those 2 subunits to then form that polymer, that polysaccharide, that complex carbohydrate. So let's now that we have done our dehydration synthesis reactions, we have formed different polymers, different types of carbohydrates, which can be branched or linear. I want you to know that its shape is going to determine its function. So I want you to quickly look at these 4 types of polymers and to yourself just describe what you are seeing in their shape. Okay, so I want you to know that the molecular structure of these polymers are not what's important. That is not going to be something that is going to be tested. What will be is, can you look at the picture, describe it, and then relate that back to its function? So now that we have talked about the structure, the function of carbohydrate, let's practice with some questions. Okay, so let's take a look at a free response question about carbohydrates. So again, pointing out the science practice it's centered around. This is over science practice number 1, that concept explanation. Again, can you recall some information that you learned in AP Biology this year? This question is framed around science practice 1.A. For you to describe biological concepts and processes. When you are describing, just make sure that you tell 1 sentence about what the particular subject is talking about. So let's take a look at the free response for the carbohydrate units. So this question says, glycogen is a complex polymer of glucose molecules involved in the storage of energy in many organisms. Describe the types of bonds in a glucose molecule. Take a moment. Think about the answer. Okay, so they want you to describe the types of bonds in a glucose molecule, and they're telling you that we have glycogen. So glycogen is a complex polymer. How do we get polymers of glucose together to form that complex polymer? Again, that's going to be a bunch of monomers of glucose, those monosaccharides that are linked together using dehydration synthesis to form that covalent bond. So the most concise answer that you can say for this question in describing the types of bonds in a glucose molecule would be that they are held together by covalent bonds. So what should we take away from today's video? The first thing is that monosaccharides are those building blocks, those monomers of those complex carbohydrates that we call polysaccharides. And that in order to put these monomers together to form those polysaccharides, we are going to have covalent bonds linking them together, again, through that dehydration synthesis to form that complex carbohydrate polymer. And then once we do link them together to form this polysaccharide, the polysaccharides can be linear or branched in structure, which would then give rise to its different functions. Thank you. Welcome to AP Biology Topic 1.5 Lipids, daily video number 1. My name is Linda Loju, coming from you from Plano, Texas. So let's see what will we learn today. We are going to learn 5 things in this video today, with the first thing being what are lipids and how does their structure affect their function. Number 2, how do saturated and unsaturated fatty acids differ in structure and properties? Number 3, how does the degree of unsaturation affect the physical state of lipids at room temperature? Number 4, what are the main functions of lipids in living organisms? And finally, number 5, what are some examples of lipids and what roles do they play in biological systems? So what is a lipid? Lipids are typically nonpolar hydrophobic molecules whose structure and function are derived from the way their subunits are assembled. So dependent on how they bond together, they're going to have a different structure, thus resulting in a different function. So let's look at some lipids. Some lipids, such as triglycerides and phospholipids, contain fatty acids. So fatty acids can be saturated or unsaturated. So let's take a look at the picture below. So in a saturated fatty acid, you can see here that there are no double bonds between carbon atoms in this particular fatty acid. You can see that it's just single bonds holding together this fatty acid. So when there are no double bonds and only single bonds between carbon atoms, we call those saturated fatty acids. Versus on the right side, if you look at that structure of this unsaturated fatty acid, you can see that's circled in green that there is one double bond. So when there is at least one double bond between carbon atoms, we're going to call that an unsaturated fatty acid. So how do these bonds affect its function? So we're going to take a look at the top picture. So in this top picture, you can see that there are no double bonds. They're just single bonds between carbon atoms. So we know now from the previous slide that because there are no double bonds, only single bonds, that top picture is a saturated fatty acid. Versus at the bottom, you can see that we have highlighted in red 2 double bonds in this structure. When we have 1 or more double bonds, we can call that an unsaturated fatty acid. So it means that double bonds in unsaturated fatty acid results in a kink in the carbon chain. The more double bonds in the fatty acid chain, the more unsaturated a lipid becomes. So the increase in double bonds is going to increase the liquidness of that fatty acid. Versus my saturated fatty acids, because there are no double bonds, they're going to be less liquid and probably be more solid at room temperature. So we have different types of lipids. We have fats, we have steroids, and we have phospholipids. So let's take a closer look at each of those types of lipids. Our fats provide energy storage and support cell function. So sometimes they can also help keep mammals warm because they provide that insulation for them. Our steroids are these 4 fused rings together. Steroids are hormones that support physiological functions, including growth and development, energy metabolism, and homeostasis. An important steroid, a common steroid found in cell membranes, is cholesterol. That cholesterol is seen, that 4 fused rings in red in that plasma membrane picture below, is essential to the structural stability of animal cell membranes. So they help maintain that fluidity of that cell membrane in the animal cells. The other types of lipids that we're going to talk about are phospholipids. So phospholipids group together to form that lipid bilayer found in plasma and cell membranes. As you can see in the picture, that a phospholipid consists of a hydrophilic head that makes it water-loving, so it will face the outside so it can be surrounded by the fluid, versus the hydrophobic tails, right? Those are water-fearing. They're going to pack on the inside to form that cell membrane. So what should we take away from today's video? We are going to take away that lipids are nonpolar. They're hydrophobic with structures that determine their function. We're also going to take away that fatty acids can be saturated, again, if they have those single bonds between those carbon-carbon atoms, or unsaturated where they have at least one or more double bond between the carbon-carbon atoms, thus creating that kink in their structure. We're going to take away that the more double bonds a fatty acid has, the more unsaturated it becomes, making lipids more liquid at room temperature. We're also going to take away that lipids serve vital roles such as energy storage, insulation, and cell membrane structure. And then our last takeaway is that examples of lipids includes fats, steroids like cholesterol and hormones, and phospholipids, which form cell membranes. Thank you so much.
Welcome to AP Biology, topic 1.6. My name is Paulette Unger, and I teach in Greenville, South Carolina at Christ Church Episcopal School. This topic will focus on nucleic acids. In this lesson, I will discuss the similarities and differences between the nucleic acid polymers DNA and RNA, and hopefully by the end of this video, you will feel comfortable describing characteristics to distinguish between these two polymers. So let's start with some things that DNA and RNA have in common. Figure 1, we have a DNA model, and in Figure 2, we see an RNA model. Both are assembled from nucleotide subunits, which are comprised of 3 parts: a 5-carbon sugar, a phosphate group, and a nitrogenous base. Each of these nitrogenous bases, when forming a linear chain, are connected by covalent bonds between the phosphate of one nucleotide and the sugar of the next, forming what we call a sugar-phosphate backbone. And each linear strand has directionality, with a 5' phosphate end and a 3' hydroxyl end coming off of that 5-carbon sugar. And when it comes to the nitrogenous bases, they can be found perpendicular to the sugar-phosphate backbone, as shown in both of these models. So when describing DNA and RNA, it's also important to look at the differences. So when it comes to some things that are different between DNA and RNA, one of those is in the sugar that makes up their nucleotides. So DNA contains deoxyribose sugar and RNA contains ribose sugar. And if we look at these two molecules, notice that in light blue, we have a hydrogen coming off a carbon in the deoxyribose sugar, but then in ribose, we have an OH or hydroxyl group coming off of that same location. So they differ slightly in the molecules that make up those subunits. Another difference is in the nitrogenous bases. Cytosine, guanine, and adenine are similar to both DNA and RNA polymers. However, when it comes to that 4th nitrogen base, thymine is found in DNA and uracil is found in RNA. So if you're looking at a visual representation of a nucleic acid and you see a U or uracil, you are looking at an RNA molecule. There are no thymine nucleotides found in RNA. Only in DNA will you see the thymine nitrogenous base. In terms of the number of strands, they also differ in that DNA is usually double-stranded and RNA is usually single-stranded, as shown here. And in the double-stranded DNA, the 2 strands are anti-parallel, meaning that they run in opposite directions, with one strand running 5' to 3' and the other strand running 3' to 5'. And if you want to learn more about what I mean by anti-parallel, you can go back and check out video lesson 1 in topic 1.5, where I discuss anti-parallel a little bit more for you. So let's practice the skill of working with visual representations and describing characteristics of a biological concept, process, or model represented visually. When you're asked to describe something, you're asked to give a detailed account of these characteristics using that visual. So let's see how this plays out with our nucleic acid polymers. So here we have a multiple choice AP question. In Figure 1, it shows a short segment of a double-stranded nucleic acid molecule. Notice it didn't tell us if we're looking at DNA or RNA, but being able to describe key characteristics in terms of similarities and differences will help us determine maybe what we're looking at. So take a moment, study the figure, highlight any key terms or labels that jump out at you or characteristics that maybe will help you select the best multiple choice answer. The question's asking which of the following statements is correct about the molecule shown in Figure 1. So hit pause, select your best answer, and then press play and we'll discuss it together. All right, so let's see how you did. Choice D is correct. It is DNA because of the nucleotides present. So let's go back to our visual and see why. So going back to the figure caption, figure 1 shows a short segment of a double-stranded nucleic acid molecule. Key descriptor here is double-stranded. DNA is usually double-stranded, whereas RNA is usually single-stranded. So that was a key difference between the two nucleic acid polymers. Another thing to look at would be in the visual itself, we see thymine nitrogen base present. And thymines are only found in DNA polymers, not in RNA. So both of those descriptors indicate that we're looking at a DNA molecule. And then when we go to our multiple choice question and we look at the choices, it'll help us rule out A and B right from the start where it's saying it is RNA. Okay, so we can rule those two out. Now we're left to choose between C and D. And choice C says it is DNA because of the nature of the hydrogen bonds between guanine and cytosine. Well, guanine and cytosine are both nitrogenous bases that are found in RNA and DNA, so that wouldn't lead us to think that this was solely a DNA molecule. However, choice D is correct. It is DNA because of the nucleotides present, since we see thymine in that visual representation, and thymine are only found in a DNA polymer. So here are some key takeaways from this lesson. Both DNA and RNA are formed from nucleotide subunits connected by covalent bonds, forming a linear molecule with two distinct ends, the 5' phosphate and the 3' hydroxyl. Each nucleotide is comprised of a sugar, a phosphate group, and a nitrogenous base. Differences, though, include the type of sugar, where we got deoxyribose in DNA and ribose in RNA. One of the nitrogenous bases differs. RNA has uracil, whereas DNA has thymine, and the number of strands differs as well. DNA has two nucleotide strands that are antiparallel, and RNA is usually single-stranded. Thank you for watching, and I hope this video was helpful to you. Welcome to AP Biology. My name is Paulette Unger, and I teach at Christ Church Episcopal School in Greenville, South Carolina. In this lesson, I will discuss the directionality of the subunits of nucleic acid polymers and how that directionality influences their structure, function, and synthesis. Then I will discuss how a change in the subunits of nucleic acid polymers may lead to changes in its structure or function. When it comes to directionality, the linear sequence of all nucleic acids, DNA and RNA, is characterized by a 3' hydroxyl and 5' phosphate of the sugar in the nucleotide. Here in figure 1, we see a DNA model, but keep in mind that a linear sequence of nucleotides and RNA would also show this directionality. DNA consists of two strands of nucleotides, and if we look at the strand to the left, the 5' end is designated here with the phosphate group coming off the 5-carbon sugar, and at the other end of this linear strand of nucleotides, we have the 3' end, which is designated by the OH or hydroxyl group coming off the 3rd carbon in that 5-carbon sugar. So this linear sequence of nucleotides has two distinct ends, the 5' phosphate and the 3' hydroxyl. Now looking at the other strand of DNA, notice that the 3' hydroxyl end is at the top and the 5' phosphate end is at the bottom. So these two strands of DNA run in opposite directions and are said to be anti-parallel. And here I've used my forearms to give you another visual in figure 2 of what I mean by anti-parallel. My forearms have two distinct ends, the hand and the elbow, much like the DNA strands do. And I've labeled my hand 5' and my elbow 3'. So when I overlap my forearms like this, notice that the 3' elbow of one forearm is opposite the 5' hand of the other. So hopefully this visual helps you illustrate what I mean when I say anti-parallel. And due to this directionality of the two strands, they're able to be held together by hydrogen bonds between the nitrogen base pairs. And there's 2 hydrogen bonds between the adenine-thymine pairs, and there's 3 hydrogen bonds between the guanine and cytosine base pairs. And the number of hydrogen bonds is significant in that the more bonds in total between the base pairs in DNA molecule, the more stable that molecule structure is overall. When it comes to directionality and function, the linear sequence of nucleotides encodes biological information. In figure 1, we see DNA in its double helix shape, and we see our two anti-parallel 5' to 3' strands. In figure 2, we see DNA unwound in its linear form, so we can really see these linear sequences on both strands of nucleotides. And each of those strands can encode unique biological information. In figure 3, we see RNA, the single strand of RNA, and its linear sequence of nucleotides, which also can encode unique biological information. And any change in the sequence of nucleotides may lead to a change in the encoded information. When it comes to synthesizing nucleic acid polymers, during the synthesis, nucleotides can only be added to the 3' end of a growing strand. So in this image, we see a growing strand of DNA, and if we want to add a free nucleotide to this strand, a covalent bond would need to form between the phosphate of the nucleotide and the 3' end of that growing strand on that sugar, forming covalent bonds. Now let's practice the skill of argumentation. It's important for scientists to present arguments or systematic reasoning to support principles or concepts or ideas. One of the tasks of argumentation is making predictions. And a prediction is a statement about what will happen or what the results might be when certain conditions are present or change. So it's an expected outcome based on your understanding of biological concepts. So what might you be asked to make predictions about? Well, if we read this task verb here, it says predict the causes or effects of a change in or disruption to one or more components in a biological system based on a visual representation. So let's practice this with the question. Here in figure 1, we see 2 separate DNA segments of the same DNA molecule. And whenever you're presented with a visual, take some time to make some observations. In this case, note any similarities or differences between the 2 segments, and then dive into the rest of the question. A scientist wants to determine whether varying nucleotide sequences within DNA molecules affect the overall structural stability of the molecule. The scientist isolates 2 separate segments of the same DNA molecule in figure 1. Each segment is exposed to similar increases in temperature within the same duration of time. Predict which segment would destabilize faster. And our task verb here, predict, is in bold for you. So take a moment, pause the video. When you have a prediction, press play, and we'll discuss the answer together. Okay, so hopefully you have an answer and a prediction handy. Let's see what we have. Segment 1 will destabilize faster than segment 2. This is an accurate prediction based on the information in the visual. Now, this particular question didn't ask for any reasoning, so this would have been all you would need to do to make a prediction. But how did I come up with this answer? So temperature is an environmental condition that can cause chemical bonds to break. And earlier in the lesson, I mentioned that the number of hydrogen bonds influences the stability of the DNA molecule. The more hydrogen bonds in total in the molecule, the more stable the structure would be. So adenine and thymine have 2 hydrogen bonds between them, and guanine and cytosine pairs have 3 hydrogen bonds. So if we look at segment 1, it has more adenine-thymine pairs and less guanine-cytosine pairs compared to segment 2. So it has fewer total hydrogen bonds and therefore will destabilize faster. Now, be mindful when you have 2 items that you're asked to make a prediction about that both are included in your prediction. It would not have been sufficient for me to say segment 1 will destabilize quickly and not address segment 2 in my prediction. Another possible prediction that would have been acceptable would be that I could have said segment 2 would destabilize more slowly than segment 1, because again, I have both of those content items in my prediction. So just be mindful of that moving forward when making predictions that have more than one item that they want you to address. You have to include both. So here are some key takeaways from this lesson. The linear sequences of all nucleic acids are defined by that 3' hydroxyl and 5' phosphate of the sugar in the nucleotide. DNA is structured as an anti-parallel double helix with the 2 strands running in opposite 5' to 3' directions. And the 2 DNA strands are held together by hydrogen bonds between those base pairs, adenine and thymine, and then guanine and cytosine. During DNA and RNA synthesis, nucleotides can only be covalently added to the 3' end of the growing strand, and any change in the linear sequence of nucleotide bases may lead to differences in the encoded biological information or the structural stability of the molecule. Thank you for watching, and be sure to check out video lesson 2, where I'll talk about protein structure and function. Welcome to AP Biology. My name is Paula Dunger. In this lesson, I'm going to discuss with you the directionality of the subunits of proteins, the 4 elements of protein structure, and how a change in the subunits of a protein may lead to changes in its structure or function. Proteins are comprised of linear chains of amino acids, and in figure 1, we see a single amino acid. If you look at the central carbon, coming off to the left, we have an amino group, NH2, and to the right, we have a carboxyl group, COOH. So we have two distinct ends to this amino acid, giving the amino acid directionality with an amino terminus and a carboxyl terminus. And as proteins are synthesized, new amino acids are added to the carboxyl group of the existing amino acid chain, forming peptide bonds or covalent bonds between the carboxyl group you see here of the left amino acid and the amino group of the amino acid on the right. As you continue to add more and more amino acids together, we get a linear chain of amino acids as shown here in figure 2 with two distinct ends, the carboxyl and the amino terminus. And if new amino acids are going to be added to this long chain, they will be added to the carboxyl terminus. This linear chain of amino acids determines the primary level of structure for a protein. And there are 4 elements of protein structure, starting with this primary level, which is determined by the sequence of amino acids held together by covalent bonds. The proteins are not yet functional at this level, and additional folding is needed. So next we have a secondary level of structure that arises through local folding of amino acid chains into elements such as your alpha helix and your beta sheet. And I'll model this for you in the next slide. But the protein still needs to have additional folding occur before it's functional. So we have the tertiary structure, which is the overall 3D shape of the protein and often minimizes free energy. So this is the more stable level of structure for proteins, and many proteins will function at this tertiary level. And the tertiary level comes about through various types of bonds and interactions, particularly with the R groups of amino acids that could vary in chemical properties based on the side chains that are linked there. Some proteins, however, do need to reach a 4th level of structure called the quaternary structure, which arises from interactions between multiple polypeptide units. So in this image, we have 3 whole polypeptide chains working together. Hemoglobin is a good example of a protein that reaches this quaternary structure. It is a protein found in red blood cells that transports oxygen. But I want to just stress here before we move on that regardless of whatever the 3D shape and functionality of that protein is, it all starts with that primary level and that sequence of amino acids and the chemical properties that each of those possess. So here in this video, I'm going to model for you the folding so you can see it all in action using a pipe cleaner and some beads. The pipe cleaner represents the carbon backbone of the primary level where you have the peptide bonds holding amino acids together between that carboxyl terminus and the amino terminus. And the different color beads represent the properties of the amino acids. So red beads are nonpolar because they have nonpolar R groups, and blue beads are polar because they have polar R groups. So next we're going to see that local folding happen. So here you see me folding it back and forth, giving rise to the beta sheet element shown there. And then we're going to have some alpha helix action happening on the right. Okay, so now we have our secondary level of structure with those 2 elements, the beta sheet to the left and the alpha helix to the right. But the protein's still not yet functional. We need to have some additional folding into that 3D tertiary shape. So let's assume that we have some water here in the surrounding environment that this folding's occurring in. The red beads are nonpolar, so they are hydrophobic, and they're going to want to fold inward towards the core of this protein. So in the core, we're going to get these hydrophobic interactions occurring since there's water in that environment. The blue beads, which are polar amino acids, they're going to be hydrophilic and interact with water. So they're going to fold towards the outside of this protein as we start to take this 3D shape. And so you see that here in our model. So here, this would be the tertiary 3D form, which could be functional for many proteins. If we want to model quaternary structure, then we need to have the interaction of 2 or more whole polypeptide chains. So here is an additional polypeptide chain interacting with our original one. So this would be quaternary structure. So let's focus, though, on our tertiary protein here. Let's say this is a hypothetical folding of a protein into a receptor protein. Receptor proteins are responsible for receiving chemical signals. The Play-Doh ball here can represent our chemical signal. The signal molecule must have a similar structure as the receptor in order to connect to the receptor. So we're going to use this hypothetical protein to practice our skill of argumentation. So argumentations are important for scientists to make, and making predictions are an important component of an argument. So predictions are expected outcomes based on changes or disruptions to one or more components in a biological system. So let's see how this might impact our hypothetical protein. So in this image you see here, let's say that the instructions for assembling this protein were changed, and this polar amino acid will be switched out for a non-polar amino acid. If we look at our question, predict the change in the compatibility between the receptor protein and the signal molecule as a result of the change in amino acid, and predict the change in the function of the receptor protein. So you're being asked to make a prediction on 2 items, so be sure you address both of those in your prediction. So I'm going to ask you now to pause the video, take a moment to think about what might be a good prediction for this question, and then press play and we'll discuss the answer together. Okay, so hopefully you have your prediction ready, and let's see what happens if we have that change. So here we're changing that polar amino acid out for a non-polar amino acid in that primary level of structure, which leads to this tertiary folding eventually. So now we have a hydrophobic non-polar amino acid that will not want to interact with the water, so it would have folded into our core, along with those other hydrophobic interactions. So in terms of the first part of our prediction, compatibility, the shape of the receptor protein will change, and therefore the protein will not be compatible with the signal molecule, as you see here. So that's the first part of our prediction. As for the second part of the prediction, the function of the protein will also be changed, and it will no longer be able to bind to the signal molecule and receive the signal, as shown in the video. So we have those 2 components addressed. The compatibility will change and it will no longer be compatible, and the function will also be changed and it will not be able to receive that signal effectively. So here are some key takeaways. Amino acids have directionality with an amino terminus and a carboxyl terminus. Amino acids are added to the carboxyl terminus of a growing peptide chain, forming covalent bonds. There are 4 elements to protein structure: the primary, which is that linear sequence of amino acids; secondary, your alpha helices and beta sheets; tertiary, your 3D functional form for many proteins; and quaternary, where you have multiple polypeptide chains interacting. The resulting 3D shape gives rise to the protein-specific function, and a change in an amino acid subunit at that primary level of structure may lead to a change in the structure and function of the protein. If you want to explore more protein folding, I've shared with you this really cool protein folding interactive found at lab.concord.org at the website address given. Thank you for watching, and be sure to check out video lesson 3, where I'll discuss the structure and function of carbohydrates.
Welcome to AP Biology, topic 1.7, proteins. My name is Linda Aloju, coming from you from Plano, Texas. In this daily video number 2, we are going to discuss the directionality of the subunits of proteins, the 4 elements of protein structure, and how a change in the subunits of a protein may lead to changes in its structure or function. So what are we going to learn in this video? We're going to learn 5 things. The first thing is what type of bonds connect amino acids to ultimately form proteins. Number 2, what are the components of an amino acid, and how do R groups influence protein structure and function? Number 3, how does the sequence of amino acids determine a protein's shape? Number 4, how do hydrogens contribute to the secondary structure of proteins? And finally, number 5, how do different interactions among polypeptides contribute to a protein's quaternary structure, and why are these structures important for a protein's function? So let's take a look at the monomer, that building block of proteins. That is going to be an amino acid. So amino acids are composed of a central carbon with a hydrogen atom. It also has a carboxyl group, it's going to have an amino group, and it's going to have a variable R group covalently bond to it. So we're going to go into these R group interactions later, but our R groups, they have properties that are similar. So they're going to have hydrophobic or nonpolar properties. They can have ionic properties, they can have disulfide bridges, and they also can be hydrophilic or polar in their interaction. Okay, now that we know the structure of amino acids, we can now put them together, link them together to make that growing polypeptide chain. So those proteins are composed of linear chains of amino acids connected by the formation of a covalent. That covalent bond is going to be called a peptide bond between the carboxyl group of one amino acid and the amine group of another amino acid. So there's going to be that peptide bond. Again, you can see in the image below in the left one, there's going to be that carboxyl group. In the right green, we have that amino group. Together, they're going to lose that water through dehydration synthesis. So as that water is removed, we can join the two amino acids together and make that peptide covalent bond to form that growing peptide chain. So now that we have that growing peptide chain, that specific sequence of amino acids and proteins will determine its overall shape. So the shape of a protein, they're going to have to first go through that primary structure, that secondary structure, that tertiary structure, and sometimes that quaternary structure. So let's take a closer look at these different levels of protein folding. So the primary structure of proteins is that simple chain of amino acids that are linked together by those covalent or peptide bonds. So just as a reminder, again, the primary structure, we're going to have one amino acid. The carboxyl of one amino acid is going to covalently bind to the amine group of another amino acid to form that covalent peptide bond that will then link them together to form this linear growing polypeptide chain. So now we're going to start getting a little bit of that dimension to the folding. So we're going to get to that secondary structure. So in our secondary structure, as you can see in the image, right, we have that peptide bond between the carboxyl and that amine group. So now what's left over is there is an oxygen in one amino acid and a hydrogen in another amino acid. So now those two are going to hydrogen bond between the atoms of the polypeptide backbone of the amino acid side chain. So those are going to hydrogen bond with each other, that oxygen and that hydrogen. Now, how it's going to bind with each other will then determine its shape. So if it's twisting around that backbone, we're going to form that alpha helix as our secondary structure. But if we are kind of folding back and forth with those hydrogen bonds, that's going to form that secondary structure of a beta pleated sheet. So now that we have the primary and the secondary structure, we can get to our tertiary structure. This is when my protein has that three-dimensional shape of the tertiary structure of a protein resulting from the formation of my different R group interactions, those hydrogen bonds, those hydrophobic interactions, those ionic interactions, or those disulfide bridges. So again, that R group interactions is going to drive the folding of this tertiary structure to give it that 3D shape. Sometimes we'll have quaternary structures. So the quaternary structure arises from interactions between multiple polypeptides. So this is more than one polypeptide chain folded in its tertiary structure to now come together to form that quaternary shape. All four levels of protein structure determine the function of a protein because, again, those amino acid R group interactions are going to determine how it folds. So let's practice. This free response question is centered around science practice number 6, predict something when something has changed in the scenario. So let's take a look at this question. It says hemoglobin is a protein with a quaternary structure composed of 4 polypeptide subunits. Each subunit contains an iron-containing heme group that binds to oxygen. The quaternary structure of a hemoglobin is crucial for its ability to pick up oxygen in the lungs and release it in tissues where oxygen is needed. Predict what would happen to a hemoglobin's ability to bind and release oxygen if a mutation caused the loss of one of its subunits. Take a moment and think about your answer. Okay, so what is this question asking us? It's asking us to predict what would happen if a mutation was there, right? So in this question 6E, there's a disruption, there's a change. So now a mutation has been introduced, which will now affect the subunits. We're going to lose a subunit. So what's going to happen if we lose a subunit in that quaternary shape? So one of those subunits won't be there. It's going to affect the way it folds. So we can say that the hemoglobin's ability to bind and release oxygen is going to be less with this mutation. So what should we take away from this video? some key takeaways. The first thing is proteins are made of linear chains of amino acids that are connected by covalent bonds, and that particular name for that, again, is that peptide bond. We're also going to take away that each amino acid has a central carbon, a hydrogen, our carboxyl group, an amine group, and a variable R group that affects its properties. We're going to also take away that the sequence of amino acids determines a protein's primary structure and then its overall shape. We're going to take away that secondary structures such as alpha helices and beta pleated sheets are formed through that hydrogen bonding around that polypeptide backbone. And that the remaining R group interactions, those remaining R group interactions is how the tertiary structure results from various interactions. Again, those hydrogen bonds, those hydrophobic interactions where they're going to want to be on the inside, that ionic interactions and disulfide bridges. Lastly, we're going to take away that quaternary structure forms when multiple polypeptides interact. So more than one polypeptide come together, and all 4 levels of structure determines a protein's function. Thank you so much.